show. Okay. So good morning, everybody. I'm Mariam. I'm a senior researcher at Eva Pharma, Egypt. And today I will present you a session about the development and the evaluation of innovative nano-based drug delivery systems for tumor targeting. First, I would like to show you the cancer in numbers based on the World Health Organization, or mainly the WHO newsroom. As we know that the cancer is a leading cause of death worldwide, and based on the data obtained from the newsroom, the WHO, you may see that in 2020, we have almost 18 million new cancer cases discovered or mainly appeared around the world, and 10 million cases of deaths around the world. And based on this data, they have classified which cancer type is the most common and which one is the leading cause of death. And based on the data, they have seen that the breast cancer is the most common cancer type, and followed by the lung cancer and the colorectal cancer, and at the end we have the prostate cancer. But mainly the lung cancer is the leading cause of the cancer deaths. Even based on the WHO predictions, they predict that by 2040 they will have around 29.5 million new cancer cases and 16.5 million cancer-related deaths annually. So this is based on how the old people behave in terms of cancer. So what about the children? They have seen that each year almost 400,000 children develop cancer, and mainly in the children, cervical cancer is the most common type. So what about the therapeutic strategies, or mainly the common therapeutic strategies of the cancer? Based on what is followed by the physicians and mainly what is found in the bibliography as well, you may see that the first line for the treatment is a surgical resection. And the aim of the surgical resection is to remove the largest or as much as you can of the tumor volume, and by keeping safe the healthy tissues surrounding the tumor. They have seen that the larger you remove of your tumor volume, the better will be the survival rate of the cancer patient. After the surgical resection, we have a certain therapeutic strategy, which is a concomitant strategy of both the chemotherapy and the radiotherapy. Mainly, they give the cancer patient a radiotherapy for 5 times per week for 6 weeks. And then they continue on a chemotherapy. I will just speak about what are the adverse effects, or mainly the side effects of the conventional chemotherapy. As you may know that most of the chemotherapy suffer from the following problems, or are facing the following issues. The first is their bad solubility. Second, they have a short half-life time. Well, they have a fast clearance, and even they don't have any targeting efficiency, like they are not targeted at all. They have a large biodistribution. And even for the radiotherapy, in order to give an effect using a radiotherapy, it has to come by a cumulative dose of radiation for a specific time. And then how can we deal with such adverse effect? So this what led the researchers to think about nanomedicine in an attempt to solve such issues. So based on the nano-based drug delivery systems, they have seen that upon loading, or mainly upon encapsulation, the chemotherapeutic agent in a nanoparticle, they can overcome the drug-limited solubility. And here, when we are saying drug-limited solubility, the drug can be either too hydrophilic or too lipophilic. Such drugs, for example, if we are facing a drug which is highly hydrophilic, so we can load it within a liposome, and if we are facing with a drug which is extremely lipophilic, so we can load it into a lipid nanocapsules. The nanomedicine, also the nano-based drug delivery system, tend to protect the drug from inactivation or biodegradation. And here we are mainly talking about the RNA, or mainly the small interfering RNA and messenger RNA, as they are mainly facing an innate immune defense using the endonucleases, leading to their biodegradation. But if we load them in a nanoparticles, so we can protect them from such inactivation. Mainly also the nanomedicine tend to extend the half-life time and even reduce the clearance of the drug, or mainly of the API used. In addition, the nanomedicine tend to provide high concentration of the drug at the tumor site, and this is thanks to the enhanced permeability and retention effect caused by the tumor, along with the size of the nanoparticle itself. In addition, the nanoparticle can help in having a sustained release effect, it can help in having like a targeted effect, in addition to overcoming as well the multi-drug resistance. So here we will shift and we will see how the nanomedicine have helped in enhancing the performance of such chemotherapeutic agents. And usually when people speak about the nanomedicine in the market, so you feel suspicious a little bit and they are worried about what will gonna happen. But let us give it a trial and let us study right now certain case studies about the nanomedicine for tumor targeting. A certain bench to market case studies. Those drugs or the case studies that I will show you right now are mainly approved by the US Food and Drug Administration as well as European Medicine Agency. So let's start with our first drug, which is the doxorubicin. As you may know that the doxorubicin, as it is mainly here, as it is shown here in the structure. Here, the doxorubicin acts as there is an anthracycline moiety, so it's an anthracycline antibiotic, and it's considered to be one of the most effective chemotherapeutics in many cancers. As you may know that each chemotherapeutic agent have a different mechanism of action. So concerning the mechanism of action of the doxorubicin, it tends to perform a certain intercalation between the base pair of the DNA strands and hence inhibiting the DNA and RNA synthesis. In addition, it tends to inhibit the topoisomerase 2 enzymes, which is responsible for the topology of the DNA and then inhibiting its DNA replication. Last but not least here, one of the mechanism of action of the doxorubicin also, it tends to lead to the formation of the free radical, causing the oxidative damage to the DNA protein as well as the cellular membrane. So what lead the researchers to think about loading the doxorubicin in a pegylated liposomal formulation is merely its a side effect. They have seen that by giving the doxorubicin in a conventional drug delivery system or merely in a conventional formulation, it is usually associated with a dose-limiting cardiotoxicity along with a myelosuppression. So what if we loaded it into liposomes? In fact, they have again with two types of liposome. The first liposomal formulation was mainly non-pegylated, which means there is no polyethylene glycol on the surface of the liposomes. And hence, it resulted in a fast clearance. And this is because when those nanoparticles are mainly found systemically, we have a certain plasma proteins. Those plasma proteins tend to undergo certain non-specific absorption on the surface of the nanoparticle, leading to the formation of a stable protein layer known as protein corona. This protein corona enhances better the recognition of this nanoparticle by the mononuclear phagocytic system, hence enhancing more its clearance or mainly resulting in a fast clearance. So in an attempt to prolong the drug circulation time and reduce the uptake by the mononuclear phagocytic system, they have added the polyethylene glycol, which is mainly a hydrophilic polymer found at the surface of the liposomes, providing not only a steric effect around the nanoparticles, so preventing the recognition of the mononuclear phagocytic system, but also resulting in a prolonged circulation time. So this is the first part. Here they ended up with a pegylated liposomal formulation of 80 to 90 nanometer size. In addition, after testing it clinically, they have seen that no signs of cardiotoxicity effect appeared on the patient, which means that there is a better tolerance of the doxorubicin dose, along with higher doses of doxorubicin can be used in the clinical practice after loading it in liposomal formulation in comparison to that in the conventional drug delivery system. Also, here by looking at the graph here, you can see that the concentration of the doxorubicin found mainly in the blood circulation and here after being infused is almost stable in comparison to the one which is fastly cleared of the free doxorubicin. Like here in the first line, which is mainly a square, you can see that after the doxorubicin is loaded in the liposomal formulation, there is no fast clearance. Okay, we have a better and a prolonged blood circulation level found in terms of the doxorubicin. But here, by looking at the free doxorubicin, it is fastly clear. And this is mainly due to its recognition by the mononuclear phagocytic system. Such a prolonged doxorubicin blood circulation resulted in a higher level of doxorubicin at the tumor tissue, and this is due to the enhanced permeability and retention effect. In fact, most of the drugs found in the market currently are mainly based on the passive targeting and not an active targeting in terms of cancer. And passive targeting means that, as you may know, that the tumor tissue is mainly characterized by its heterogeneous nature, and that the vasculature over there is mainly a leaky vasculature. We have a discontinuous pericyte layer. Even we have for the tight junctions, there is no good tight junctions found over there, resulted with a fenestrated vasculature. Such a fenestrated vasculature enhances more and more the penetration of the nanoparticles. In addition to that, we have for the very poor lymphatic drainage within the tumor environment, which results in the enhanced retention as well of the nanomedicine over there. So it's a way of, as you may know, a benefit from the nanomedicine side due to its small size, and another benefit from the tumor side due to an enhanced impermeability retention effect. The second case that we will study right now is mainly the Paclitaxel. As you may know, that the Paclitaxel is a class of the taxane anti-tumor drug. The mechanism of action is that they are a microtubule stabilizing drug that tend to disrupt the microtubule dynamic, leading to or mainly inducing the mitotic arrest, leading to a cell death. As you may see here in the chemical structure of the Paclitaxel, it is mainly an extremely hydrophobic molecule. So how this drug will be mainly given intravenously for the conventional formulation present previously. They have tried to add the Paclitaxel in addition to the cremophore, which is mainly a non-ionic surfactant beside the ethanol. This was in an attempt to improve the Paclitaxel solubility. Well, they have seen that after adding the cremophore, it resulted, or mainly the patient suffered from three severe side effects. First, neurotoxicity, neutropenia, and hypersensitivity. So it is not easy at all, which led the physicians and researchers as well to advise with a pre-medication with the glucocorticoids along with antihistamine drugs in an attempt to reduce the hypersensitivity caused by the cremophore, as well as reducing the Paclitaxel dose to be administered to the cancer patients. So in an attempt to remove the cremophore from the commercial Paclitaxel formulation, they have tried adding such a hydrophobic molecule in either a polymeric micelle or even in a human serum albumin-bounded Paclitaxel nanoparticle. So here we will just study the case of the human serum albumin-bounded Paclitaxel. It has a size, or mainly hydrodynamic size of 130 nanometer. So they have seen that after loading the Paclitaxel in the human serum albumin bounded particle, it has completely removed the cremophore from the formulation as a whole, resulting in no hypersensitivity, no neutropenia, no neurotoxicity, and hence there is no need to have a pre-medication with either a glucocorticoid or mainly a antihistamine drug. In addition, and instead of having a limited dose due to the cremophore, since the cremophore is removed from the formulation, so there is a greater administrable dose of the Paclitaxel as well. In addition, The Paclitaxel itself, after being bounded with the human serum albumin, as you may see here, that the size of the particle is 130 nanometer. Here you may see a very good sentence, a very good graph, presenting that this large particle disintegrates into small human serum albumin bounded paclitaxel complex having a size of 10 nanometer after being immediately administered intravenously. Such a complex is merely entered the cells using a caveoli-mediated transcytosis. Such an uptake mechanism also helped in a better uptake of the paclitaxel drug inside the cell, rather than having only a paclitaxel which is not albumin bounded. Even here, by looking at the graph, you will find that the paclitaxel, after being bounded with the albumin, resulted in a faster tissue distribution and merely a faster blood clearance. And this here is an advantage, and it is not something bad, because after being exposed to a large concentration of a paclitaxel systemically, this could result in a systemic toxicity. So thanks to the nanoparticle, which is merely to the complex itself of the albumin bounded to the paclitaxel, which results in the shorter duration of high plasma drug concentration and mainly diminishing the risk of systemic adverse effect resulted by the paclitaxel. The third case that we will study right now is the vincristine sulfate. The vincristine sulfate is mainly a potent chemotherapeutic agent. Well, and its mechanism of action is mainly the microtubule inhibition and arrest of cell division in the metaphase. Mainly the side effect of the vincristine sulfate when taken in the conventional drug delivery system is the neurotoxicity. And usually the effective dose of vincristine sulfate in the cancer is mainly a 2 milligram per meter square of the body surface area. But due to the neurotoxicity caused by the vincristine sulfate, this resulted in a dose capping, ending up with 1.4 milligram per meter square of body surface area only to be administered to the patient. So here we have a dose capping problem. In an attempt to overcome such a dose capping issue, they have loaded the vincristine sulfate in liposomes. The composition of the liposome is here a little bit different. It's mainly composed of sphingomyelin and cholesterol-based liposome, having a hydrodynamic particle size of almost 115 nanometer. And this was in an attempt to overcome the dose capping. I just would like to add something here. As you may know that the liposomal composition is not usually the same, or mainly it is not usually standard to have the same liposomal composition for every API. It mainly depends on two things. The first thing is that what is the compatibility, or mainly what is the physicochemical properties of the API to be loaded in a liposome having a certain composition. Second, the release, the in vitro release, or mainly the in vivo release of the drug inside the liposome itself. So it's a matter of matching which composition of the liposome will end up with the good results after loading the API. So here, thanks to the liposomal formulation, which ended up with a vincristine dose intensification, after facing the problem of the dose capping, we have returned back to giving the normal dose of the vincristine sulfate, which is mainly 2 milligram per meter square of the body surface area. In addition, the liposomal formulation lead to a better penetration and concentration of the vincristine inside the tumor tissue. In addition, it resulted also in a prolonged half-life time and reduced the clearance rate of the vincristine up to 100-fold. So here we have studied several cases. One case about the dose capping, another case about the toxicity, and here I will show you another example of a nanoparticle. And it was mainly marketed as the first marketed nanomedicine for dual drug delivery in case of the acute myeloid leukemia. Here's a little bit unique type of nanomedicine, as it contains mainly two APIs, or mainly two drugs, which is mainly the daunorubicin and the cytarabine. The daunorubicin is mainly an anthracycline, but the cytarabine is a nucleoside metabolic inhibitor. Here I will just go directly to the therapeutic strategy of the acute myeloid leukemia. The standard care for it required the patient to take the cytarabine for 7 days and the daunorubicin for 3 days, which is 7+3 regimen, followed by either a consolidation chemotherapy or stem cell transplantation. Such a therapeutic regimen is modest clinical benefit, and this is mainly due to the overexpression of the MDR-related proteins, such as the P-glycoprotein in the patient with the secondary amyloid leukemia. So, in an attempt to overcome such the modest clinical benefit, they came up with such an innovative idea, which is why not having a synergistic effect by adding 2 different drugs in a different ratio, which is mainly the composition of bilamellar liposomes having a 100 nanometer size, hydrodynamic size, containing cytarabine and daunorubicin in a 5:1 molar ratio. And here you'll find that both drugs are, by looking at the chemical structure, are hydrophilic. So they will mainly be present within the hydrophilic core of the liposome itself. Such a bilamellar liposomes containing 2 different APIs with different ratios resulted in a maximum synergistic anti-tumor activity. In addition, by looking at this beautiful picture, you will understand the benefit of adding such a composition or mainly such synergistic drugs within the liposome itself. Mainly the daunorubicin, which is mainly of an anthracycline chemical structure. After being administered in the tumor microenvironment, or mainly after being in phase with the tumor microenvironment, resulted from the degradation due to the efflux pump found over there. But after administering the daunorubicin inside the liposome itself, this offers a sort of protective effect to the daunorubicin from this efflux mechanism, and so improving its half-life time. So the second one, which is for the cytarabine itself is facing as well a sort of degradation due to the cytidine deaminase enzyme, but after being loaded in the liposome itself, it resulted in a sort of protection as well from the degradation. And so the mechanism is very interesting, and such a synergistic drug delivery system is mainly after being loaded in the bone marrow, result in a sustained release of both the cytarabine and daunorubicin in a very protective way from the normal degradation mechanism found over there. And so here, if you look, that we have a prolonged exposure of the CPX351, which is mainly the cytarabine daunorubicin 5:1 in the bone marrow, and sustained delivery of cytotoxic drugs ensure the death of the tumor cells. So, which is really an interesting example for a synergistic effect or mainly synergistic anti-tumor effect. So here I would like to show you the picture of some drugs, mainly from the bench to market. So now I don't think that we have to worry or mainly to feel frustrated about having or suspicious about the nanomedicine in the market. Already we have lots of drugs found in the market for composed mainly of nanomedicine for tumor targeted. As you may know, the Markepo, the Ampizom, the Doxil, Abraxane, Univide, the Vexus, Myoset. So each of them is a liposomal formulation or mainly a PLGA formulation of a certain anti-tumor drug. Here also, I would like to show you a table containing the product name, which is for example, doxorubicin, the API name, which is doxorubicin, the composition of the nanomedicine itself, the particle size, what is indicated for, and the manufacturer, and also the approval year, whether from the Food and Drug Administration or from the European Medicine Agency. So as you have seen that it is sort of, or mainly it is safe, it is a safe drug delivery system which helped us to overcome several side effects produced by the conventional drug delivery systems. Since or mainly the examples that I have just shown are mainly about a passive targeted cancer nanomedicine. So here I would like to show you an example of the first active targeted cancer nanomedicine, which is still under clinical trials, which is mainly a docetaxel. The API here is a docetaxel. It's mainly loaded in the PLGA or mainly pegylated PLGA nanoparticle. Mainly when we say that it is an active targeted cancer nanomedicine, it means that the surface of the nanoparticle itself is composed of a certain ligand. Those ligands are mainly specific to a certain sorts of receptor overly expressed on the tumor cells itself. So here, our ligand is an anti-prostate specific membrane antigen, which is the PSMA targeting moiety. Such a targeted moiety tend to recognize the receptors that are mainly overexpressed on the surface of the prostate cancer cells and the tumor associated neovasculature, because such a neovasculature are not only fenestrated and having a very bad tight junctions and very, very bad microenvironment, but also there are certain receptors overly expressed on their surface from which this antigen can easily bind. And here, the clinical activity of the bind, the 0114, in both the anti-prostatic specific membrane antigen and the undetectable tumor types showed that it can bind in a very good way to such receptors overly expressed on the tumor cells, in addition to the APR effect normally produced by the cancer itself. Based on the bibliography that I have read, and based on what the researchers are trying to prove, or mainly are trying to say, the leading mechanism in any anti-cancer therapy is mainly due to the enhanced permeability and retention effect. And here, thanks to the fenestrated vasculature of the tumor, which enhance more the permeability of the nanoparticle, and thanks as well to the poor lymphatic drainage found inside the tumor environment, which enhance better the retention. It comes after that having an actively targeted cancer nanomedicine. And this is a little bit difficult because not all of the cancer cell will have the same type of receptors being overly expressed on the cell itself. It will be very, very, very specific to target such a receptor overly expressed on the cells. So here, as you may say, despite that the bind 0114 is the first active targeted cancer nanomedicine, still the APR effect was the greater driver of the drugs of this drug to come in contact with the tumor microenvironment, rather than the ligand, which is more and more mainly specific to the receptors overly expressed on the tumor cells. So here we are done with some of the drugs that are mainly in the market and some which are in the clinical trial phase. Here, what about the recent nano-based drug delivery systems for tumor targeting? I know that some teams in the US are mainly working on a golden nanoparticle using the green nanotechnology. Mainly from a point of view, it's mainly safe using the green nanotechnology, and from other side, using the golden nanoparticle is a sort of a new trend and a promising therapy for the cancer itself. But also, what about using the messenger RNA and the small interfering RNA for the tumor targeting? As you may know that the messenger RNA is mainly a single-stranded RNA, and when we use it as a tumor targeting therapy, the aim of the messenger RNA is just to overexpress the protein suppressing tumor. So here, for example, they have loaded the messenger RNA in the PLGA polymeric nanoparticle. Well, in an attempt to or merely to overexpress the protein, leading to suppress the tumor itself. So here they have tried using a mouse model. As you may see that the first mouse are mainly injected using a phosphate buffer saline, which is mainly and only a buffer. There is no API found inside it. Here, they have only injected, in the second row, they have only injected the messenger RNA only without being loaded in the nanoparticle. And in the last case, they have injected the mouse using the messenger RNA loading inside the PLGA nanoparticle. So usually having a messenger RNA naked without being loaded in any nanoparticle will lead that this RNA will be more exposed to biodegradation using the RNAs and endonuclease. So in an attempt to protect it from such degradation, it will be better to load them in a PLGA nanoparticle or in whatever sort of nanoparticle based on its properties. So here at the end, even by looking at this picture over there, you will find that we have a reduction in the tumor size itself. Even having a polymeric lipid hybrid of the messenger RNA nanoparticle itself resulted in a micropinocytosis uptake mechanism, which is a little bit different from having the messenger RNA only uptake mechanism, which means that by having the micropinocytosis due to the PLGA nanoparticle loading inside the messenger RNA has mainly protected the messenger RNA from the biodegradation. Here, for the small interfering RNA delivery, it suffers from the same problem as the messenger RNA. But here, mainly the small interfering RNA is not that much used as the messenger RNA, and even in terms of the bibliography, you will find that we have a lot of numbers of the messenger RNA bibliographical studies in comparison to those of the small interfering RNA delivery. As you may know, that the small interfering RNA is mainly composed of the 21 to 25 base pair, and we have two strands. We have either a guide strand or a passenger strand. It merely, the mechanism of the small interfering RNA is based on the formation of the RISC complex, or merely RNA-induced silencing complex. And one of the two strands, which is mainly the guide strands, bind to Argonaute 2 protein, leading to the formation of the RNA-induced silencing complex, and hence target a specific region within the messenger RNA, leading a gene knockdown. So here, as you may see, that they have also added the small interfering RNA within a oligoarginine functionalized pH responsive polymer. So here, the type of the nanoparticle is a little bit different because it's mainly responsive to a certain stimulus within the tumor microenvironment. You know that tumor microenvironment is a little bit highly acidic. So here, they have used this polymer to be in response to the tumor microenvironment, in addition to containing the RNA inside it. And even after testing, I want to show you something which is very interesting over there. Here they have compared both what about having a naked small interfering RNA and what about having a nanoparticle loading with small interfering RNA. You have seen here that the injected dose over time is almost stable in comparison to that of the naked small interfering RNA, which means that a nanoparticle has mainly protected the small interfering RNA from the biodegradation. Even the same upon testing its effect using the animal model, injection at first with phosphate buffered saline, we have controlled nanoparticles, meaning which are nanoparticles which are not loaded with any small interfering RNA. We have here the small interfering RNA only, and in last year we have the small interfering RNA loaded inside the nanoparticle itself. You have seen that here we have the blue color means that we have a lower tumor size, but the red color means that we have a high tumor size. You see that when we have the phosphate buffer saline only, there is no difference, there is no reduction in the tumor size. Same for controlled nanoparticles. there is almost no difference. But here, upon comparing both the small interfering RNA alone and the small interfering RNA load in the pH responsive polymeric nanoparticle, we have here a better reduction in the tumor size in comparison to the innate one. So thanks to the nanomedicine technology, which helped us to overcome such an adverse side effects, or mainly adverse side effects in terms of the conventional drug delivery system, and offer a protective effect in terms of the RNA technology. So here, in conclusion, we can say that the nanomedicine has provided several advantages over the conventional systemic administration drug delivery systems, as we have seen in the doxorubicin and in the Paclitaxel examples. Nanomedicine were found to be effective in modifying the pharmacokinetics and the biodistributions of the bioactive agents, while mainly decreasing their systemic exposure and toxicity. But you may know that nothing is 100% perfect, and we have usually to take care of certain properties, or mainly certain factors, while designing our nanoparticles, if our aim is to target the tumor, or mainly in an attempt to improve the nanomedicine's tumor accumulation, intratumoral distribution, and even the intratumoral retention. First, you have to take care of the size of the nanoparticle, as mainly having a size from 1 nanometer till 10 nanometer will mainly enhance more the glomerular filtration, and so you will have an extremely fast clearance of your nanoparticle. So the best thing is to be, or mainly to have a nanoparticle ranging from 10 nanometer to 100 nanometers, to ensure that it will have a better intratumoral accumulation based on the APR effect. Second, what about the surface feature? As you have seen in the doxorubicin example, when they have just added the polyethylene glycol on the surface of the nanoparticle itself, it has better enhanced its physicochemical properties in terms of clearance. Such decoration with a hydrophilic polyethylene glycol resulted in a steric effect, meaning, or in other means, it has protected the nanoparticle from the non-specific protein absorption, or mainly protein corona, and so it has resulted in a less uptake by the mononuclear phagocytic system. Third thing, you have to make, or mainly you have to make sure about the nanoparticle stability. You have to make sure that your nanoparticles are stable. So you have also to take care of their stability in vitro, their stability in vivo, and even about their storage stability. Last but not least, here you have to take care about the nanoparticles interaction with the tumor microenvironment. Based on my master studies, we usually say that you have first to study what is the tumor microenvironment in terms of the pressure found over there, in terms of the drainage found over there, in terms of the acidity or mainly of the pH found inside it, so that you can better design a nanoparticle which could lead to a better accumulation, even better targeting within the tumor itself. So here are the references of what I was just presenting you. I'm sorry not to add all of the references, or I mean not to add each reference separately in each slide, as one slide could contains like for 5 or 6 references. So I prefer to add them all in one slide, so that it will be easy for the one who would like to revise the slides or have a look at them later on, to have an easy access to those references. Here I would like to thank my supervisors found at the Munir Armanios Research Center by Eva Pharma. As you may know that being a young researcher is not something easy at all, and even working in the research field is something which is a little bit not easy as well. So it's usually great to have a very nice and a very good supervisors. And I was usually taught that it is easy to find a job, it's easy to find a PhD position, it's easy to find even a postdoc offer, or to find whatever you want. But it is not easy to find a very good supervisors doctors who usually tend to support you, encourage you, and help you. So in this occasion, I would like to thank Dr. Laila Gadrab, Dr. Basim Sami, Dr. Mina Amir, and Dr. Joseph Boulis for their usual support, effort, and time mainly during my practical work in their laboratory. So thank you, and it was a pleasure being with you today and presenting such a talk about the nanomedicine and how the nanomedicine, we can see them from bench to market, and the beauty of the nanomedicine and the intelligence of the nanomedicine, and how the nanomedicine have saved several adverse side effects caused by the conventional drug delivery system. Thank you.
Thank you so much for your presentation. Floor is open for the questions. Anyone have any questions?